It's a real pleasure for us to welcome Professor Mashiraka today for us. Monash. I think it's fair to say he's one of the leading scientists on the dendritic computation in the world. And based on his very interesting findings, he has developed a theory called dendritic integration theory, which is trying to explain the consciousness experience. Unlike other theories of consciousness, which typically assume that neuron as just a binary switch, but anyone who had studied neurons know that neuron is much more sophisticated, just more than a binary switch. It's more intricate, and it has very interesting dynamics in itself. And what I like about Matthew's theory is that it's based on his finding on neuron, its physiological characteristics, which I really like about it. And today I think we can hear more about his theory. So please welcome Dr.
Thank you. Very kind introduction, and if anything about that is true about me being leading dendritic physiologist, then it's because I'm standing on the shoulders of giants, and one of the giants is in the audience, Greg Stewart, who put down, I think, the path that I followed when I came to the same institute in Heidelberg. I arrived in 1997 or 8, I can't remember, which was soon after Greg had just left and really made his mark. And I think in a very really true sense made a lot of what I'll show you today possible. So I'm going to talk about dendritic integration theory, which is probably an overblown description of the let's say a whole lot of converging lines on what's going on in the main neurons of the cerebral cortex during sensory perception, and with a link to well, in some sense, I think we truly are working on conscious perception in the sense that the animal is conscious and it's perceiving. But I think that's where we have to draw the line, because we can't ask the animal what it's feeling. And so it's not about subjective ideas of consciousness, but the difference between being awake and not awake, or aware and not aware. In any case, I think it's a theory that distinguishes itself by being very low level and trying to guess at high level things. And I'm part of a consortium that has just published a paper, a sort of overview in this paper of 5 different current theories of consciousness. And I think it's very clear, I think, at least from the ones chosen in this paper, that this DIT, as I call it, dendritic integration theory, is really going bottom up. And at some point, I think it's true that we stop almost where some of them start. But hopefully it's still informative and converging and so on. That's the sense in which it's overblown, is that I don't think we're getting anywhere close to some of the things that people would want to call consciousness at this point. And nevertheless, I do think that it's based on not just findings that I think you can take to the bank, but also a lot of converging lines, which makes me also feel that I get ever more confident we are on the right track. Anyway, I've tried to summarize here. There's a lot of text, I know, but I've cut and pasted that from this paper, and I'll quickly go over this so you don't have to actually read it and work out what all that text means. So I'll quickly just go through, and essentially what I'm saying is this was the 6 observations that led us to suggest that there should be a bottom-up theory like this. And just really quickly, when I came to Heidelberg, I literally took over where Greg Stewart had left off in terms of recording from the dendrites of these really big neurons, the layer 5 pyramidal neurons of the cerebral cortex, which project out of the cortex and also throughout the cortex, and they're ubiquitous across the whole cortex. And what you're looking at here is a cross section of the mouse cerebral cortex, and you can see, because in this case there've been only the layer 5 neurons are expressing a visible fluorescing compound or protein. You can actually only see the layer 5 cells here, but you can see that basically it's very uniform and distributed, and it's basically ubiquitous everywhere. And just to cut a long story short, the finding that I found interesting back then, this is the end of the 90s, we named backpropagation activated calcium spike firing, and the point about this, if I can, I don't think I can actually. Wait, I've got to get my. Give us it back. I need my cursor to be able to start this. What you're looking at here are two recordings from the same neuron, and one is on the top of the neuron and one is on the bottom of the neuron. Oh, it's just stopped it. Yeah, here we go. And the point is that there's a current injection at the bottom and a current injection at the top, meaning that we're kind of faking input to this cell. One is a fake input that you might be considered a bottom-up or feed-forward input that's causing an action potential in every iteration. And the other one is a moving but faked, if you like, compound EPSP that could be thought of as what it might look like if feedback came to the top of the cell. And then I hope you can see that there's a special moment in this protocol where the two combined gives you this amazing effect, which is the combination of a backpropagating action potential. Here we go. There's the backpropagating action potential that you can see is backpropagating because this red version is the reflection of that in the dendrite, and that literally was, I think, one of the most groundbreaking events in at least in my time at the time, which came out in Nature in what is it, 1995, I think, or it's 1994, sorry, from Greg, and was the reason that I wanted to go to this lab and look at it. And people wanted to know what the point of this was, and this seems, in my opinion, to be one of the points of the backpropagating action potential, namely that it can somehow potentiate the events that go on in the dendrite. One of the events that goes on in the dendrite, which was also discovered in collaboration with Jackie Schiller by Greg, is a so-called dendritic calcium spike. And the point here is that it's quite, you would think it's quite unlikely to get this dendritic calcium spike because there's a high threshold for generating it in the dendrite. But combined with the backpropagating action potential, it's not so high. And so that turns this into an associative device. And it can basically, you can, I think from that point on, you can think of this as a two-compartment neuron. I don't know if it's hearing me, yeah. So it's got one compartment at the top, if you like, or one dendritic tree, which is receiving input, and those inputs are predominantly feedback inputs coming from various different regions, and one compartment at the bottom that's predominantly feed-forward receptive field, if you like. And so one way of interpreting the whole point of the architecture of the cortex, and the reason why you have these ubiquitously distributed layer 5 cells and so on, is that they're combining these inputs. And they've got an intrinsic mechanism that's built into the ion channel distributions in the cell that allow them to detect when there's feed-forward and feedback in coincident input to the cell. So that's the one thing. And this immediately makes us think of the feedback being not just important, but somehow intrinsically built into the cortex. Along the line, I like this idea from Anil Seth of perception being a controlled hallucination. And essentially, this was early work done in vitro, which I just showed. And I spent some time starting up a lab in fact in Switzerland, and then moved back to Germany and to Berlin where I could start a lab where we were really focusing then on trying to see if there is any if there's any larger meaning to this mechanism. So my trajectory, my scientific trajectory, has been a quest to see whether or not there's any cognitive manifestation of this really biophysical effect at the cellular level. And one way of looking at this was to go over to an age-old paradigm, the perceptual threshold paradigm. Here, when it's done in humans, this is a very common form of it, where you do some somatosensory stimulation, and you do EEG on the subject, and basically the idea is to see what if there are conditions or you can do this such that the subject doesn't feel it, so lightly that it's not felt, and then you can also do it above the threshold for perception. Is that me? No. And above the threshold for perception, you'd like to know. Essentially, you're asking what goes on in the cerebral cortex that distinguishes the subliminal stimulus from the liminal stimulus. And so for a long time, we've known that there's a distinct event that happens in the EEG. It's a delayed event. It's not related to this first volley of information, which is the feed-forward signal. And it's been long interpreted that this would be some kind of feedback signal in the cortex. But of course, that's more or less as far as you can go with a human. And it was then done in monkeys, and in monkeys using inserted electrodes, you can actually, I think, be fairly convinced that this is indeed a feedback signal. It's something to do with the upper layers of the cortex and so on. So that's a position where we were in for a long time without being able to make any more progress. When I had a really talented postdoc in my lab, Naoya Takahashi, who's since left the lab, but he did some beautiful work, which I'll just really quickly summarize, on the perceptual threshold in mice now with whisker stimuli. And it's essentially the exact same idea. We move the whisker. Well, first of all, we train the animal to perceive a whisker stimulus and lick for a reward. And secondly, we then change the stimulus intensity, and we try to find the psychometric curve that describes the reaction of the mouse, and we assume that that's perception, and that by definition in this case the perceptual threshold is the 50% detection rate, or in other words, in that case, that's for us is the lick rate. When at that particular stimulus strength, it licks half the time and it doesn't lick the other half time, and we consider that the time that the rat is perceiving this, I suppose, consciously. Although again, we can't ask the rat, so that the report is the lick. Good. And our hypothesis here is that when it's sub-threshold, we don't get much feedback, or it's not triggering this mechanism in the dendrite, and with top-down feedback, it triggers this mechanism, and that's somehow related to the perception. So that's where we started. That's where we hypothesized. We wanted to test that, and it turns out that that's not so hard to test. Luckily, partly because this signal in the dendrite is calcium, and we've got lots of ways to test that. And these days we can do that optogenetically. And secondly, it's also quite standard now to use 2-photon imaging, which allows you to go right into the cortex. It's easy to record at this relatively superficial depth in the dendrites of the pyramidal neurons. And you see these little dots here. This is basically a plane that's at this level of this pink line here, and you can see lots of dendrites. Each dot is an apical dendrite of a pyramidal neuron, and we can record the calcium changes there and ask the question: do they correlate with the moment of perception, namely with something going on around this level of stimulus? And so there's so this what just popped up is stimuli at exactly this intensity at the 50%, and you can see that because half of the trials, which are each stripe here, are misses, which means the mouse missed to lick at the right opportunity, and approximately half of them are hits. So we're at the perceptual threshold, if you like, and this is a heat map, so you can see the dendrites. We picked one particular dendrite here, and it's predominantly showing a calcium signal when the animal licks, and that's the average of all of the hit trials and then missed trials. So there's a correlation there, but that's not causation, and we wanted to go a bit further than that. I don't think yes, so for the correlation part, we started manipulating the ability of the dendrite to cause a calcium spike, and we either inactivated the dendrites or activated the dendrites. And in the case that we inactivated the dendrites, it shifted the perceptual detection curve or the psychometric curve to the right, and in the case that we activated the dendrites, it shifted it to the left. And here, perhaps remarkably, this is actually the zero value, so where there's no stimulus at all, the animals licked at, I think, about 40% lick rate. So you could possibly consider that a hallucinating a stimulus. In any case, it established, I think, beyond most measures of doubt, the fact that there's some causal relationship between what the activity of the dendrites and the perception of the animal. And lastly, which, yeah.
Have you ever tried no report paradigm in this situation where you present the stimulus as a much smaller threshold?
Yeah.
And then not give an opportunity for them to report. So basically, if they are like human, they are definitely perceiving things, but not reporting.
No, we haven't done that. And I guess you also mean like use some feature like the pupil dilation and so on.
Yeah, maybe some kind of a control measure may be necessary, but I'm just curious if you don't ask them to report or give no opportunity to report.
Yeah.
Then if all these activities are related to the act of the report itself.
Oh, I see, because they have to lick. No, we did some things along that line. We didn't exactly do that, but we were looking at, for instance, the delay to lick, which is informative in terms of which component is responsible for the signals in the dendrites. And there's things going on in the dendrites before the actual movement of the tongue.
But you have catch trials that you can...
And we have catch trials as well. Yeah. So, well, yeah. Good. So the last thing, which was the third thing on the first list I showed you, was that we wanted to know which kind of a pyramidal neuron it was and where it was projecting to. So because these neurons are obviously affecting something downstream. And it turns out there are two kinds of layer 5 pyramidal neurons. There's the so-called intratelencephalic or IT neurons and pyramidal tract, or more commonly these days, extratelencephalic neurons that project out of the telencephalon and go to various subcortical regions. And it turns out we can get transgenic mice for either of these neuronal types and ask this question separately. Cut a long story short, it turns out to be the PT neurons that if you block all of these neurons, you'll get a decrease in perception in about 45% of these neurons, 46% of these neurons are contributing, if you like, to the perceptual detection. In IT neurons, it's much less. So they're not totally uninvolved, but absolutely less than the PT neurons. And then we could go further with this, and since this was a chemogenetic approach, meaning we express receptors in these neurons that we could block, or we could activate so that it inhibits the neuron, and the inhibition in that case works with another ligand, so a chemical that you put wherever you want. So we could put the chemical in all of these downstream regions that projects to, and the upshot of that was that the main region that is important for the projection here is this region of the thalamus, a higher order thalamic region, which for somatosensory cortex is called the posterior medial nucleus of the thalamus, but we presume would be true of all higher order regions of the thalamus in terms of according to modality. It was also actually we also saw effects if we blocked the superior colliculus, and we saw effects if we blocked the striatum. The biggest effect was if we blocked the higher order thalamus related to the somatosensory loops. And we didn't see any change in the psychometric curve if we blocked the brainstem on either of these regions. Yeah.
So these two classes of pyramidal cells are defined here by their outputs, where their axons project, but a lot of those, the thalamic regions and stuff also have reciprocal connections back to this. Do you know, do the inputs going into layer 5, do they diverge among the two cell classes, or is that?
They go to both.
They go to both.
Well, actually, it's good that you asked that right now. I'm coming to a fork in the road at the end of this talk if I don't go too slowly. And I thought that this would be answering the longer answer to your question would involve one fork in this road. So we come, and I think it would be interesting to. So it turned out that Christoph Koch and Francis Crick had thought about this question a long time ago. They kind of rejected this possibility afterwards, but for a time they were suggesting that the large pyramidal cells in layer 5 that fire in bursts and project outside the cortical system might be the neurons that whose firing correlates with awareness, and they thought it would be marvelous if that was true, but unlikely to be as simple as that. And I guess that this is a paradigm in which it's probably it could be as simple as that. But actually, I think that there's a lot more as we explore this that needs to be said in any case. But anyway, they were suggesting it back then, so that's if you like some step forward. So that's now focusing. I've just gone through these first 3 observations. Now I want to go through the next 3 observations, which is basically relates to a sort of chance discovery related to anesthesia, which is a big topic and one that we kind of stumbled into. We have to anesthetize our animals at various points during the experiment anyway, so it was kind of destined to happen. We were I don't want to waste too much time going into that, but basically we was probing we wanted to ask the question, what's the causal influence of the top of the neuron in general on the bottom, so it's very on topic, and it's turned out that with transgenic animals and optogenetics, this turns into an easy question. You can express light sensitive channeled opsin in these neurons. I presume I don't need to go in depth here, but just ask me if you need more information. And we can, using what we call a periscope, so a light guide with a mirror at 45-degree angle, we can shine light horizontally. And that's fairly easy. It's like doing extracellular recordings, basically, once you've got the transgenic animal set up. And so we can definitely excite just the tough dendrites of these neurons. And then we can do other experiments like recording the activity in these neurons, or do population recordings and so on. So that's the configuration of the experiment. And the other thing, of course, is recording from the cells. So one way to record from the cells is to do what technically here is a juxtacellular recording, but effectively is a single unit recording from a defined neuron. And if you do that, you get what we expect actually, which is burst firing from this cell, given what I showed you earlier, that you get a calcium spike presumably, and that causes the neuron to fire. And so we were kind of astonished when we anesthetized the animal to find that it flatlined. And this is all the more astonishing if you've been working in vitro, as I had been for 2 decades or 3 decades up to this, no, not quite 3 decades up to this point, because essentially the claim here, so the light is opening channels in the membrane, and so current's going in, and the anesthetic, which is very unlikely to block channel opsin, and in fact we could subsequently prove doesn't, is therefore causing a signal every time in the top of the neuron. So for it not to have any influence on the output of the neuron, the anesthetic has to be somehow preventing the signal from getting from here to there, and that opens a whole new vista of possibilities, and speaks to, I think, given that we anyway think that there's some sort of correlation between what goes on here, the activity up here, and perception of the animal. It immediately made us think, well, I wonder if that's related to the mechanism for anesthesia. And so we looked further into that. And basically the idea would be anesthesia is somehow decoupling the top of the neuron from the bottom of the neuron. And one could imagine that this is happening ubiquitously across the whole cortex, and that essentially you're cutting off feedback essentially through this mechanism that therefore can't go subcortically anymore, can't return, if you like, to subcortical structures. Now, that's the other thing that we knew in advance and from other people's experiments, that if you block the thalamus, that also has a huge effect on consciousness. In fact, some people's theories of anesthesia involve this site of origin, if you like, of action of the anesthetic to be in the thalamus, and therefore that somehow has some effect on consciousness. We could test this by actually deliberately blocking the thalamus, and by blocking the outputs of the thalamus onto various targets back in the cortex. So first of all, blocking the thalamus, and now is that a question? Yeah.
So about the previous figure, what's the kind of anesthesia, and also does it depend on the dose of anesthesia?
Yeah. So the first answer is mostly isoflurane. But we also test it with urethane and ketamine xylazine. And we're currently in the process of going back to other anesthetics like propofol and probably pentobarbital. And well, the other thing was the dose response, and
it's related to that ketamine
Yeah?
specifically.
Okay.
Because the ketamine, a small dosage seems to induce hallucination in human, right?
Yeah. So we haven't done the dose response curve yet, but there's a person doing that in my lab as we speak, hopefully. And the issue with ketamine is really interesting, because previously, some time ago, back in Switzerland, we were looking at the effects of anesthetics in vitro, and we also used ketamine in those experiments. And there was a very counterintuitive response to ketamine, which is generally thought, the action of ketamine is generally thought to be the block of NMDA receptors, but it's a very dirty drug with lots of side effects. And in our case, although we saw effects that seemed to indicate that the calcium spike was reduced in vivo by ketamine and other drugs like urethane, and there it was pentobarbital. In vitro, it was the opposite. So the other anesthetics reduced the calcium spike in the dendrites, but ketamine wildly increased the calcium spike in the dendrites. So it remains possible that one explanation for the hallucinogenic effects of ketamine is that in a dose-responsive way, you get some upregulation of the calcium spike and a downregulation of the NMDA spikes, and for some window of ketamine, you can get, let's say, hallucinations or overregulation. That's one explanation. That's all we know so far. I can't tell you that that's actually the truth.
In the previous slides, you mentioned that PD neurons project to some of the thalamus, and I was wondering if this projection from cortex to thalamus, how is it related to the signal coming from thalamus to the cortex?
It's a similar question to one before. How is it related? Well, certainly it's going directly to particular neurons that loop, and in some cases loop right back to the same neurons that are being stimulated. I think in general, I think it's getting clearer and clearer, not just from our experiments, but from lots of experiments, that the thalamocortical loop is really intrinsic here, and it's a loop. It's not just the feed forward or what was projection down from the cortex. so there's a more precise answer, which I was also saving up in terms of what it's actually doing. So maybe if we just wait till the end, and maybe I'll definitely go down this pathway. But that's the general thing, is that we want, there's some a loop that needs to be established. The thalamus is downregulated by anesthesia. We can deliberately downregulate the thalamus and see what happens. And before I go there, and this maybe triggered your question. One thing we know about the return loop from the higher order thalamus is that it doesn't just go to layer 1, which is astonishing enough, but it also goes to layer 5A, and it's not insignificant in its projection there. And I don't think anybody really knew what this projection does. And we're only just trying to get on at the moment to understand what the projection of layer 1 does, and layer 5A was not known. But one of the things that we could predict, I think, was from Murray Sherman's work and other people's work, is that the higher order thalamus activates, I think, about 50% of its action goes through metabotropic receptors, not through ionotropic receptors, and that's an opportunity for us to pharmacologically distill this as well. And so, putting a drug in thalamus that downregulated the thalamic response also had the effect of downregulating or uncoupling the effect, meaning when we shone light on the distal compartment up here, at the same time that we were blocking the thalamus, the control, which is red, was reduced to the purple here at the cell body. So blocking the thalamus reduced the effect of light, affecting the soma or the cell bodies of these neurons, which is quite a circuitous route to go. I should just point out there that it didn't block the effect on the dendrites itself. So that's one of the many ways in which we ever showed that this is not an effect on the actual signal in the dendrite, but an effect on the propagation of this signal, the influence of this signal. All right, and we also could do this for the various drugs that blocked both metabotropic glutamate, and it turned out also metabotropic cholinergic receptors. Both of those had an extraordinary effect on the ability of light to activate the bottom of the neuron. So this closes this loop, if you like, and I've wasted so much time getting to this point that I almost can't go on for what I really want to show you. But basically, these are the 6 observations that led us to want to call this a theory, if you like. Because they're all convergent. It does now fit, I think, with the suggestion that the thalamocortical loop is really intrinsically important, and these neurons are really intrinsically important for both establishing and maintaining the thalamocortical loop. And so, and that the feedback that goes to the top of these neurons is all part and parcel of that, of maintaining that loop. And so that's all self-consistent. It doesn't tell you why that should be linked to consciousness. And so in that sense, I started by saying, we've brought it so far from the bottom up. Not that I think there are really good explanations at the moment. It's not like we're the deficient ones and everyone else has got an explanation for that. But we just press on at this point, and that's not quite, but nearly as far as we've got. This is essentially another group that was doing very similar experiments in monkeys, showing how important the thalamus was for controlling the thalamocortical loop. It's just one example, but it's an interesting one that came out not so long ago. And our claim is basically this on the left, that there's some nexus point in the apical dendrites of pyramidal neurons that is being maintained by thalamic input, such that if you block that, you'll lose this nexus point. But when you do that, it basically separates this link has no influence. Basically, it can't push the thalamocortical loop, and therefore you break the thalamocortical loop. That's our best guess at why that brings together all of these disparate findings. And so, in some sense, you could say nature built in a switch, which is this very important nexus point for transmitting the signals. Let me go really fast through this. And so this is a new topic in the lab, and since we're almost at the end, I'll just show a couple of slides. This was the alternate route that was going. Because we've recently been looking at this layer of the cortex called layer 6B. Some people call it layer 7. Cajal called it layer 7, but I think most people don't like admitting that the cortex has 7 layers. So it gets called, I think, mostly 6B. And we've been working on it now for several years. I have a really talented postdoc, Tim Zolnick, who's been doing this. And I won't get into how he got interested in these neurons, but there's a thin layer of neurons at the bottom of the cortex. And what's interesting, first, there's 2 really interesting things, both with relation to the input to these neurons and the output to these neurons. First of all, they only get cortical input. In fact, they're the only layer in the cortex that doesn't get any thalamocortical input, strangely enough. That's slight exaggeration. You can just barely detect some input to this layer, but it's really, really small and out of proportion to any other layer. And on the other side of the coin, the output from these neurons, astonishingly, goes to exactly the same layers as the higher order thalamus, namely layer 1 and layer 5A. And in this case, it also projects to the thalamus. So it doesn't get input from the thalamus, but it does give output to the thalamus. And actually, if you look even more closely, I don't think I've put the data in there, but the projections to layer 1 and layer 5 are to the same sublayers, layer 1A and layer 5A, as the higher order thalamus. So it seems to be very, very specific targeting, just like higher order thalamus. And to cut a long story short, because I don't want to spend all afternoon on this, but just to whet your appetite, we've done a bunch of experiments on what the function of this particular layer of cortex is. I can't skip over the fact that they've got lots of orexin receptors, and orexin is related to sleep-wake cycle and arousal, and so there's every chance that this layer is related at least to arousal. In this particular experiment, we took the sleep-deprived animals and then head-fixed them. They were transgenic animals in the first place with chanorodopsin it's a transgenic animal for a subpopulation of layer 6B neurons. So they're expressing chanorodopsin and therefore are excitable. And then we could put light on the column with where these on a particular column that we are recording from with a silicon probe, and we could observe the behavior of the animal. And when you see the blue dot, that's when the light comes on. You can see just after this, it starts to whisk and continues to whisk. But if you look at the EEG, or the local field potential, in these recordings, what you see is the typical delta during the non-stimulating phases. And then for the 1 second of photostimulation, you see this sudden transition to high gamma, and then a sudden transition back to slow delta, which doesn't actually look like what's going on at the behavioral level, although it's very hard to know. We don't know at this point whether it's awake or not. It's got a kind of awake-like EEG. We do know that that's not entirely local, because we can move the silicon probe to more distal regions, and you do see, albeit less of a high gamma, but still enough to show that there's a sharp transition in these other regions. But nevertheless, we don't know what to call the behavioral state of the animal at this point, and that's sort of ongoing. So that's the collected results showing the changes in the different wavelengths with respect to awake and with the photostimulation. So it indeed has a really powerful effect on what you might consider the thalamocortical loop, if it's the thalamocortical loop that's the most important thing for wake versus sleep. And I think in this context, what one should imagine is that you've got this thin strip around the cortex, at the bottom of the cortex, which is effectively a second thalamus. It's a second thalamus that gets only cortical input, but nevertheless impinges on all the nodes of the thalamocortical loop, and not just the nodes in the sense of this neuron and that neuron, as in layer 5 cells and higher order thalamus, but the two nodes, the two compartments of the cortical neuron. And so I think that's, again, another, from my point of view, convergent evidence that we are indeed on the right track for looking at what's important for awareness and so on. Another way of looking at this is that it's a mechanism for attention. So essentially, there's some other cell type or mechanism to impinge on the thalamocortical loop. And I was sort of alluding earlier to wanting to, I'd like to hear your opinions on this, because in some sense, it's always, I think, or for long been suggested that actual consciousness or awareness in the general sense is different to attention, that they're two different mechanisms. And I guess we would like to suggest the same thing, that this may be some mechanism you've got to, let's say, control your attention or your awareness in some sense, that is subliminal, that you don't know about, or at least you only have loose control of, or it doesn't, let's say, percolate to your consciousness. Whereas the other one is the moment of perception. I'm thinking at this point, well, one suggestion from Tim, and which I think plausible, needs to be tested, would be that something like a gist, when you can't remember somebody's name and it starts with C, but and maybe it's a long name, and you maybe could say some other things about it. And in fact, it's surprising all the things you can say about it without knowing what it is, and then suddenly it pops into your mind. That's so you could imagine that you're searching with some sort of process that is activating different cells, basically testing to see whether that hypothesis works. And when you get a hit, it's because you engage the thalamocortical loop, and there then you're in. But of course, that's all hand waving. I think that would need to be further done. But going along the lines that Niall's already explored, it would be the perfect mechanism to suggest as the dichotomy between attention and awareness in that sense, or at least one way you could conceive of that.
That's very interesting proposal, and I just want to make sure the meaning of this yellow line. Does it imply anything about sort of the connected kind of network across all the areas of the cortex?
Yeah, I took this diagram because I guess there's a whole lot of what we say, suggestion here, subliminal suggestion to. I guess I'm assuming off the bat that in the end, when you are aware of something, it's because you're engaging the right thalamocortical loops that are very specifically pulling out the right ensemble of neurons or columns or whatever in a very specific pattern. And that would constitute any given percept would be the right combination of this. And one presumes that the thalamus is doing this somehow, is picking out the right columns. And if that's the case, then I want to put something else on top of that can do this, the initial search part of that. Well, maybe I'll back off from that. The anatomical and physiological, the functional anatomy would suggest that you've got a system that can impinge on that, so that if you like, the thalamocortical substrate supervenes on this layer 6B higher kind of.
I see. So because I wanted to understand what you meant, but is it correct that you are suggesting like thalamocortical loop as a kind of vertical thread, and this layer
I basically wanted to get a little bit away from, yes, it's kind of vertical in the local, but it's got some
and the horizontal.
It's got some configuration. Yeah, I imagine that you're only probably dimly aware of a really tight thalamocortical loop in any given area, although enough to perceive a whisker stimulus apparently. But but I guess that when you, in the larger context, you're linking this to all the other things that should happen in any given perceptual task, and you want to, there'll be multimodal, there'll be also the other, by the way, this is one thing I struggled with all the way through writing this recent review with all the other theories, if you like, is that everybody else asks up front, are you suggesting that consciousness is at the front of the brain or at the back of the brain? And is this a yeah, I mean, for people who haven't come across that idea yet, it seems extraordinary that would be the question. But there's also the idea of whether the distinction between phenomenal consciousness and access consciousness, which kind of divides like this, where the phenomenal consciousness would be, let's say, the experience that you have that is whatever it feels like to see red and so on. But then there's another kind of consciousness, which is the way you control this, if you like, and your ability to report it and so on and so forth, and your introspection on this and so on, the way you can manipulate feelings and so on, which is generally thought to be something to do with access consciousness. I'm quickly getting into territory where they'll all jump on me at this point and say that I've got their theory slightly wrong on that. But the nice thing about the DIT, I think, is that it's agnostic here, and it just says, well, you've basically got the same architecture everywhere. You've got these pyramidal neurons everywhere. So it's going to be one and the same thing in terms of coupling across pyramidal neurons. Exactly how that manifests and what it feels like, I don't really have a comment on, but basically that would mean that you don't have to have, just in the same way we don't have to have the argument as to whether or not the consciousness is generated by the thalamus or by feedback in the cortex. We don't have to have the argument as to whether it's at the back of the brain or the front of the brain. It's just basically, or at least we can tie it down, if this is really on the right track, down to some mechanism that's easy to see and easy to deal with and easy to talk about, so that we can get on with experimenting with it and so on. So very quickly, because I know I'm really over time now, is that I'd skip all through what I would advise you to read about, just because I want to get to some final wrap up. Where we think that it's not just that you perceive in the moment, but that you store associations. Essentially, that this connectivity, what we here are calling context, but basically represents the other activity in the brain that's not related to whatever this feature column is computing, but only in the sense that it correlates with it, or associated activity in the rest of the brain. That this is stored here in the sense that you learn to expect from the strength of the connections here and the context, what else would be going on in the brain when one particular feature is activated in certain different conditions. That's what you would call associations or context. It'd be somehow the closest thing to semantic memory, and we think it makes sense that it would be in this region of the neuron, and we've got, I think, very good evidence that it is there, but I won't go into the evidence. But essentially then, all the other structures that we tend to call memory structures would then should be, in that paradigm, be considered gating structures, which basically give you criteria for when you should change the strength of the contextual inputs to here. And that would allow me just to wrap up in another sense. I know that's just that's the 30-second version of what takes usually a whole talk, but just so that I can get to this this wrap-up slide, the idea would be, I think we should be looking at this neuron not just as two compartments, but three compartments, i.e. the apical compartment, the basal compartment, and a coupling compartment, which is really important, and that corresponds actually roughly or quite closely to the dendritic arborizations. So there's an apical dendritic arborization, there's a basal dendritic arborization, there's so-called oblique dendrites along the coupling zone. And again, that seemed to be important for some reason, and it's hard, I think it's been hard to say what that is. Well, we would be claiming that they are controlling the flow of signaling both up and down the dendrite. In this kind of a context, the output of this neuron is doing some sort of Bayesian computation that's telling you the probability of this feature given the context. And in this context, you want the input to the bottom of the neuron to be quite stable, meaning once you've learned, presumably during some critical window where you learn about the statistics of the world, it shouldn't change too much. And then the other one should be, and that would then ground your your world, as it were, in the way that you've wired up your cortex. And the other kinds of inputs would be more labile, or should be more labile. You should be able to change your associations very quickly. And, you know, 1 minute this means lick, and the other minute it means don't lick, or whatever. And that should be quite labile, and should be and would be sensible to put in a different region of the neuron, and to have it only targeted by contextual input, and so on and so forth. It would make a lot of sense to this. And in some sense, this would be the content then of your memories. And if you want to go the whole way, it would be the meaning of some inputs. And lastly, from the anesthetic results, we'd basically be saying some input that's coming from higher order thalamus controlling this region is controlling your ability to be aware because of the thalamocortical loop, but also your access to the memories, the conscious memories, if you like. This would be the kind of memories that are associational or semantic, would only be accessible if the top of the cortex is signaling the bottom, or the top of these neurons is signaling the bottom. So with that, I've got so many people to thank, but in particular Naoya Takahashi and Moritaka Suzuki, who I'm only mentioning right at the end, but he did all of the work with the anesthesia. It was an amazing set of experiments that he did, and then the people involved in the memory experiments that I only just alluded to, and of course Tim Zolnik, who did the work on layer 6B. Well, thanks.
Question from here, but there's some question in Zoom. So from Razban, how many types of neurons are in layer 6B, and what are the regions which doesn't have a layer 6B?
Good question. So there are currently two transgenic animals, the so-called DRD1A transgenic and the CDGF, that get overlapping populations of excitatory neurons. The CDGF one targets a smaller population that goes only to layer 1, and the DRD1 has 3 populations, one that goes to layer 1, one that goes to layer 5A and the thalamus, and another one that goes to the superior colliculus. And they represent combined only 50% of 6B. And so there's another set of neurons, and they're mostly inhibitory in layer 6B, and that seems to be for signaling within layer 6B predominantly, and a little bit into layer 6A. And I didn't get to show all the results of Tim, but getting back to the 2 questions that were asked about the specific connectivity here, the layer 6A goes entirely to, well, it goes in its projections to layer 1 and layer 5A. It targets only pyramidal neurons, layer 5 pyramidal neurons, not layer 2, 3 pyramidal neurons, not interneurons, not anything above layer 5, or even layer 6 in that sense. But it does target both the ITs and the ETs, so the intratelencephalic and the extratelencephalic. The DRD1 neurons, which are a bigger population, so that's the ones that go to the subpopulation of layer 6B that goes to all of these 3 regions, layer 1, layer 5A, thalamus, and the superior colliculus. It also targets some interneurons in layer 6, but and it avoids all the excitatory and pyramidal neurons in layer 6. So it's got really specific targeting, which is another smoking gun. I think that it's really alone, it's just remarkable. I think that it can avoid. It's got all the tough dendrites of layer 2, 3 neurons, which are actually much more numerous than the layer 5 neurons, and it avoids all of those and just synapses onto layer 5 cells. So then going on, Tim did an incredible amount of work. So I sort of direct you to this paper of his, because I have trouble remembering all this information, and so you certainly won't on one go through. But in addition, he looked at how the thalamocortical loop comes back, and whether or not it's the same neurons that are being picked out by the so it's 6B to layer 5, back down to thalamus, back into the cortex. And as far as he can work out, they seem to be very likely the same neurons. So they're picking out, if you like, there's got some sort of specificity at the single neuron level here. The higher order thalamus, now finally to answer your question, goes also to layer 1 and 1A and to layer 5A. And there it has differential effects from the 6B. So 6B causes, lo and behold, an event called an NMDA spike in the tough dendrites of layer 5 ET neurons, which is profound. It's almost feel like I should show you, but anyway, I ran out of time. There's not enough to say all of this, but a profound influence on the generation of little explosive bombs in the thin dendrites at the top of the neuron, which are just about as powerful as a calcium spike. And it also has strong effects on the oblique dendrites. If you now compare that to the higher order thalamus, the higher order thalamus also projects to the ET neurons. It's not as powerful as the 6B neurons projection, either to layer 1 or to layer 5A. But it's still causing nonlinear events that are NMDA dependent, or that are blocked by APV. And then if you can continue this long functional anatomy lesson, because you can also ask, how does this work on the IT neurons, the intratelencephalic neurons? And there it's slightly reversed. It's the comparative influence of layer 6B and higher order thalamus is switched, such that they both have fairly strong influences. They're both, except in one case, NMDA dependent, NMDA receptor dependent, but the higher order thalamus has a stronger influence on the intratelencephalic neurons. What this all means, I don't know. But one thing is that the other thing to say is that the thalamic inputs are generally depressing, and that means that if you give a train of inputs, the first one's bigger and the second one's smaller, and in the train they all decrease. The layer 6B inputs are generally facilitating, which is the reverse. They get bigger and bigger in a train of action potentials, and they're not just facilitating in that sense, but they're, I don't know the next word to use, but something like augmenting. If you did the same train, if you wait, 5 seconds and then do another train and then wait 5 seconds, do another train, the beginning of the train is bigger and it gets bigger again. So they're getting bigger and bigger, and they're facilitating in each train. So this suggests, I think, or maybe I suggest, I hypothesize at this point that the thalamocortical loop is somehow precarious, that if it's hard to maintain. It's really important. You don't get consciousness without it, but it dies away if you don't maintain it. And on the other hand, this 6B mechanism is constantly feeding into this loop in a really forceful way, and it can regenerate the thalamocortical loop. So you could just about imagine at this point that you need the two to work in tandem to maintain a train of consciousness, if you like, or a train of thought, and that it would be simultaneously maintaining thalamocortical loops and deciding which thalamocortical loops are more important than not. So again, that's all hand waving. I only know the functional anatomy and everything I showed you, but that's one sort of speculative way you could go. I saw your hand up, Greg.
So what happens if you inhibit these sites?
We're just in the process of trying that out at the moment. We were doing this chemogenetically, and the first chemogenetic experiments, in a scary way, were causing all sorts of lung embolisms and so on in the animal, and we're just redoing those experiments. So I don't know the answer, but yeah, we could speculate that this will turn the animal, either give it narcolepsy, which, by the way, is something that you get if you block orexin receptors, and orexin receptors aren't just in these neurons, so that doesn't close the story there. But anyway, it could well cause narcolepsy, or you could get something like a zombie, where the animal is effectively not perceiving I say zombie because I don't want to know. So that gets back to the larger question here as to whether we're pushing at the mechanism for anesthesia, or whether this is just the icing on the cake of anesthesia, and this is just the link between the cortex and the subcortical structures, but not the core or whatever, that was the woo sound, of consciousness, where exactly and what we should attribute to consciousness there. So it's possible that we just take away, let's say, the ability of the animal to use its simulation box or whatever this cortex thing is doing. Yeah.
I was wondering whether you could try and relate your very first slide to some of the later perceptual work. So the idea of separating just spikes and spiking rate and calcium and bursts of spikes, and how they separately relate to.
Was this my first slide? This one?
Yeah. So
okay.
Can we relate the presence of those bursts or otherwise to like sensory coding and the perceptual consciousness?
Well, that's what we're suggesting.
But have you?
I don't know what more you want to
perceptual
get.
experiments, so you must have the bursts, and you must be able to analyze them
So
separately.
actually, there's a finding that's kind of gone under the radar, I think, which is if I'd given the larger talk on the memory part of this, would have got to something that I think's gone under the radar, which relates to, I think, the question of are bursts somehow special here? And it's been really frustrating in Naoya's experiments. He's the one that did the perceptual detection experiments. We also looked at we also did multi-unit recordings and looked at the bursting of neurons, and frustratingly, the calcium spike was discriminative in a statistical sense, and the bursting wasn't. Mind you, the bursting was actually get more cleaner data from the calcium spikes than you do from multi-unit recordings in layer 5. And so it's possibly in the statistical noise here. But I saw some experiments recently from Mark Harnett. I don't know if you know him. He's another guy in the dendrites field, and he's just done recordings in awake and anesthetized, looking with the neuropixels. Should I explain? And so he's got hundreds of recordings from all depths of the cortex, and he asked the question whether bursts were special, and kind of disappointingly sees more or less the same bursting from every layer of the cortex, not just layer 5. And when he tries to block the specifically calcium spikes in the layer 5 neurons, in his terminology, had no effect. In what I saw as data, which was just from a really recent talk that he gave, I saw a 20% effect as far as I could eyeball it from his. So it wasn't no effect. Now, the under the radar experiments where we did juxtacellular recordings from layer 5 pyramidal neurons, and we were teaching the animal basically to lick in response to the stimulation of its own brain. And then we went back in, we grabbed one neuron by the scruff of the neck, and we could fire it, and we could ask, does it change the probability that the animal will lick? So we've got one neuron, and we can do this either as a burst or as a regular spiking. And lo and behold, bursting was salient and not bursting wasn't salient, which I think is the first and only, dangerous thing to say, but anyway, if this is categoric information or evidence that bursts are more salient. And so they are kind of special. Why? But it's not easy to detect this, is the other corollary, that you're really close to the noise. So why this should be the case, I don't know. It could be that certain bursts in certain neurons is important, or maybe just at certain times the bursts are important, or so on. So it's frustratingly hard to prove this. And as far as I know, I know of people working in monkeys, just trying to think of the name now, who think they have evidence that the bursts aren't important, and it's difficult. I'm going to guess
I think
that
it's hard to separate because you have a higher probability of a burst when you have a higher firing rate.
exactly.
So
Yes, it's very hard to disentangle this. By the way, it was discriminative. Just firing rate was discriminative, not the bursting. But the burst is a small component of the high firing, as you were just saying. So that's very frustrating, and it could lie in some circuit where it targets some burst in some neuron, goes to goes to a higher order thalamus that gets a special projection back, as seems to be implied here, and therefore all the other bursting is probably important for other reasons, but not that particular reason.
I'm curious on the fact that there are certain type of inhibitory neurons that affect or can control the apical dendrites.
Yeah.
And I'm wondering if anyone done any experiment to specifically block those and get the same effect in terms of blocking the apical dendrites and then see the same decoupling. Yes,
that's quite right. So of those, very prominent amongst the neurons you're referring to are the somatostatin Martinotti neurons in particular that project from deep layers to the calcium initiation zone in the apical dendrite, but also layer 1 NDNF neurons or neurogliaform neurons, which seem to work through a different mechanism, a GABA B mechanism, and they're both really effective in blocking calcium spikes. And we've done one of those two experiments where we excite the Martinotti neurons, and they have huge effects in all of the domains that we were showing. One of them was, I don't think I showed that, or did I? I can't remember if it was on the slide. No, I left it out. But one of the other manipulations we did was, was channelrhodopsin in Martinotti neurons. That was another way we could inactivate, and that had a huge effect on the perceptual detection curve. So yes,
I'm just asking this question because, like, it's always known that parvalbumin interneurons are the one that change the rhythm, gamma oscillations, all these things.
Yeah.
But if only somatostatin can do this or neurogliaform can do this, then where do they have this effect, the parvalbumin neurons?
Well, so it seems to be. So first of all, parvalbumin is kind of ubiquitous through the different layers, and they seem to be to some extent controlled by VIP neurons that also control the other neurons, and I think the idea, or the current speculation, is that the parvalbumin neurons are doing some sort of important gating or timing in a general sense of the, say, the oscillatory nature of the activity, or at least let's say timing, and that these other ones are doing specialized functions, and possibly mostly to do with the active properties of dendrites and so on. And in that context, you could imagine that whereas one is you obviously there are two, let's say, parallel or maybe orthogonal mechanisms in that scenario where one is gating the timing so that you're catching the right rhythms, and the other one is switching off a particular class of events. And to close that loop, the VIP neurons are disinhibitory in both instances, so that they're basically stopping the other interneurons from doing what they should do, and that I think the corollary of that question, and that my answer is that there should be some mechanism to activate VIP neurons if ever you want to be conscious, because otherwise if you buy this whole DIT, then you'd have to switch off the inhibitory neurons that are otherwise preventing dendritic events, and so that would come with it.
Can I ask one more question? Your last slide, that you had this idea that the apical dendrite are just sort of context versus the basal dendrites that do the feature. Yeah. So is there any anatomical evidence of differential plasticity between the one that is context specific versus the one that is feature specific?
Oh, that's a great question. That was actually another topic of Mark Harnett's recent talk, and it's active investigation at the moment. And there's another phenomenon. I don't know if it's probably going off on a tangent. There's a lot of what that particular workshop was about was a new phenomenon called behavioral timescale synaptic plasticity, BTSP, which Jeff McGee's been working on, but now a lot of people are jumping on board. And Mark Harnett was asking the question you're asking, which is because this now relates to whether you can get that kind of plasticity at the bottom or the top of the neuron. And he was going with extracellular stimulation, which is very specific to starting at the bottom of the neuron going up. And to my surprise, he found the opposite. He found something kind of like this plasticity in the basal dendrites, but not as he went up, although he only went up sort of halfway. So I don't know the answer to the top. I'm going to guess. Well, I didn't have a lot of time to dwell on it, but this. Oops, where have I got my cursor? Let's see. Yeah, this is, we think, working somehow like a switch, and it implies, I think, that there's either some cofactor or some other neuron that it's, you know, some intermediate neuron or something that this does that decides whether or not this other connection is being regulated. And therefore, I can also imagine this is one of those really difficult problems for neuroscience, and would explain why no one's seen it yet, which is that under normal circumstances, you wouldn't get this, let's call it the factor X, whatever factor X is that's being delivered here, is not normally present under any normal in vitro conditions that people look for plasticity in. And so you're not finding that plasticity. And so the thing that we call LTP or other kinds of plasticity is the thing you get without that factor. That's one possibility, let's say. Oh, I think Greg was.
Just to comment on that question.
Sorry. Yeah.
Just to comment on that question. I
Yeah.
mean, firstly, people have observed plasticity of the inputs onto the distal dendrites.
That's true. Yeah.
Of these neurons, so it does exist, and people have also observed that the plasticity rules for plasticity on distal dendrites is different from proximal dendrites. So the spike time independent plasticity rules are different, and that's work that I've published, but also Michael Häuser has published.
Right.
So there are differences.
Yeah. Thanks for that. Yeah, mind you, the kinds of changes that I would have been alluding to here, although I think it's up in the air at this point, but I'm imagining that the changes you have here relate to the susceptibility to activation of NMDA spikes in this region. Of course, that could just depend on the size of the inputs, but it might also relate to the excitability of the tuft. But I agree with it.
Now? Yeah, good. So yeah, I have been wondering, and this is more like a high level question, and just that you mentioned this X factor of what's happening there. How do population dynamics come into the picture? And is it possible that maybe it's something about multiple neurons and like regions and hubs of or networks of neurons that actually add this memory switch into this, into like a certain single neuron?
More than likely. So I guess I imagine that the point, one of the issues going on with this mechanism, I think, the one that I alluded to here, is that since it's a coincidence mechanism or associative mechanism, you could imagine that you've got two different streams of information that are overlapping, and that automatically means that only some of the neurons that get this stream and some of the neurons that get this stream get selected out, but that seen over a population, that's a very precise ensemble of neurons that essentially are getting well, depending on which words you want to use here, the data that is necessary to say that let's say this feature should be recognized, and the context to say it's likely. And I could have used several different combinations of language there, and in the end, that's just language. But probably better to bring this back to the biophysics that I would predict from the biophysics that you should expect really precise activation of ensembles based on the particular overlap of the the one pathway and the other pathway. And I think for me, one of the pressing questions to answer at this point is whether or not either or both of these pathways is itself very selective, or whether one or other is selective and the other is broad, let's say. So you could imagine a situation where, let's say, the feed forward is selecting very precisely neurons that should be getting data, but there's a kind of broad distribution of contextual input, and that it's therefore getting connections that way. Or now, having looked at the Layer6B connectivity and its real precision here, I really wouldn't be surprised if the feedback is also very precise. And the other thing that lends to the suggestion that it's actually very precise is there's a whole story about NMDA spikes, which I didn't go into at all, but an NMDA spike is predicted from a cluster of input. That's to say synaptic inputs that are close in time and space on the dendritic tree, which means that even if you had, let's say, broadly distributed input, that's a contextual input, it could be that it only meets in a cluster on certain dendrites, and therefore is specific in that sense. So it's going everywhere, but here it's just sporadically synapsing, but on one particular place you get a convergence. And I think that would make a lot of sense. And actually, NMDA spikes have been seen in the basal dendrites too, so you could invoke that kind of specificity at both ends of the neuron. And then you would get an additional kind of specificity, which would be, where's the coincidence of these things going on? So I guess in the final analysis, that would be my prediction. But there's quite a lot of work that needs to be done to show that. Mike Heuser recently had, I think, the first demonstration in vivo of a really specific feedback input actually causing what looks like an NMDA spike on the distal tufts. But that was really mind-blowingly difficult experiments that it's going to take some years, I think, to get there.
Just about the, thinking about completely different species, like, lacking this thermocortical loop, like for instance, fly, they can sleep and, be awake. Do you have any kind of speculative idea how, the sleep and awake cycle are similar or different between species?
Yeah, I mean, so all the work's been done in mammals, and it looks like in a mammal, this is a really important system for controlling cognitive space. But when we come to this larger question of just general awareness, which could relate to something that's more closer to, say, arousal, then I think we should expect subcortical structures to be very much involved in arousal. And I mean, if for instance we could treat the six-layered cerebral cortex as like some people think a simulation box, basically a prediction of what's going on in subcortical regions, then that leaves open that there are kind of more general principles about arousal that relate to subcortical structures, and that your simulation box is just interfacing with this, and that without your simulation box, you're still aroused and you're still, quote unquote, awake. Now, what that would mean, that the thing, I think the whole field of consciousness is basically weighed down by the baggage of first person perspective here. I mean, it's much easier to look at whether an animal's aroused and in that sense awake, than it is to ask whether the animal is experiencing something at any given time. And I think there's good evidence from humans and perceptual detection experiments and so on that the six-layered cortex is involved in being aware in a perceptual sense of things. So it doesn't mean that because that's the case in humans, that's also the case in animals. It also doesn't mean that you're not aware in some sense when you're not aware of, let's say, subcortical happenings going on. And in point of fact, the very fact that you need this thalamocortical loop is already a suggestion that there is some really hard and fast link between cortical and subcortical, even in mammals and even in humans. that's to do with that. My guess is that in the end, when we start going down the phylogeny, that it'll turn out that it'll be easy to find principles of arousal that go across species that relate to subcortical structures. I don't have any firm prediction about how this is going to go when we start ramping this up to higher and higher cognitive events, and I'm not sure if I should care about what we call this the experience of a fly or versus the human. But I don't know. I don't have any other good comments after that.
Can you take the question from John Rand from Razvan?
It's a message to you already.
So Razvan asks, how many cells need to be manipulated to get the shifting perception threshold? I think that's one of the experiments.
Well, we actually don't know. I'd love to know the precise answer to that question. Although we could. So the only experiment where we played with the numbers and we've got down to 1 was kind of, well, I suppose it's related. We can train a mouse to lick if you stimulate its brain, and in this case, there's a distinction between what this is actually a whole body of work done by Michael Brecht in his laboratory, and he's shown that if you do what he calls microstimulation, which is an extracellular stimulus of the cortex, it's very easy to get the animal to lick in response to that. In fact, way easier than any other kind of paradigm that you would use. And then you can go and take one of those neurons, and one of those neurons will reliably make the animal lick with higher probability. So I should have put that the other way. It will unreliably make the animal lick, but it's statistically significant. And I could guess from, who asked the question? Was it somebody online? Oh, sorry. I could guess from that you might expect the perceptual detection to change with just one neuron. Certainly, it's not unthinkable, but we haven't done the actual experiment.
These lines, and also it brings other factors like learning and stuff like that into the picture, especially about the role of L1, for example, you said in the context. But like the work done by in Randy Bruno's lab, like especially removing layer 2, 3, and including layer 1, and the animals are still able to detect stimuli, or earlier work, I think, by Hobson in the 80s, they remove somatosensory cortex.
They the whole cortex, yes. So that's a good question, and good topic to bring up. I was kind of alluding to this, that it might be that I don't know where to jump here, that it seems that you need the cortex to do things that are difficult or new or somehow unexpected. A need in quotes that you're much better at let's say dealing with unexpected things and new things and so on with a cortex. But it also appears that you quickly get, in a sort of Kahneman way, very good at something which isn't necessarily cortical. And certainly in mice, if you overtrain them, you don't need the cortex, and you can remove the cortex. Or if the task is sufficiently difficult, you don't need sufficiently easy, you don't need the cortex. So my guess is that this thing that probably in humans is really, really important is only less and less important as one goes down the phylogenetic tree, if you like, and that that it's kind of a good thing if you don't have to think about something. And so from an evolutionary perspective, it's actually advantageous to funnel everything to subcortical structures if you can reliably predict it, and it's very important, then you'd like to do it quickly without thinking about it. But if it's something that's changing, or that you know needs some that isn't obvious, or is even counterintuitive, then you need this special structure. That's my best guess as to how to interpret the Randy Bruno experiments, where he's taken out the cortex and nevertheless this animal behaves perfectly okay. I think if you look at those experiments, first of all, they were very, very simple detection experiments. Secondly, it is an effect that's in the first day you get some, you do see some effect of taking out the cortex. So that speaks to it being important in the beginning to have the cortex, and not so much in the end. But I think it's very easy to develop, even in mice, tasks for which you absolutely need the cortex.
Thank you very much for the talk and also the participation from audience. And we wrap up. Thank you.
Thank you.